A pleasant day to everyone. It is an honor and a pleasure to participate in a discussion on climate change and an exploration on how molecular biology can assist in our journey towards mitigation and adaptation. My name is Chana Camille Vinci Cruz Abeledo. I'm a teacher, a researcher, and a content creator. I come from a seemingly diverging background, from molecular biology to environmental science and management. It's not exactly a straight path, and I would like to share my experiences with you at the nexus of molecular biology and environmental science. Molecular biology is the subfield of biology that studies the structure and function of the macromolecules, such as proteins, lipids, carbohydrates, and nucleic acids, that are essential to life. It is often mistaken for microbiology, which is the study of microscopic organisms such as bacteria, fungi, and viruses. But molecular biology is about what happens inside the cell, the interplay of the different structures and the molecules that make them work. More often than not, when people think of molecular biology, they imagine the sterile working conditions of the laboratory. And a lot of this is true. In order for us to understand how the cell works at the molecular level, we do need the sterile and controlled environment present in laboratories. However, this doesn't mean that what we do is not connected to the greater expanse of the world and the environment. After all, the stimuli that triggers the different reactions of the cell come from the environment, and the information encoded within the molecules inside our cells help us adapt better to environmental conditions. So, how does molecular biology help in our environmental and climate change efforts? Well, let's first talk about biodiversity. The link between biodiversity and climate change has long been established. Throughout Earth's history, the climate has always changed, with ecosystems and species coming and going. Rapid climate change does not give time for ecosystems and species to adapt, and so biodiversity loss increases. Estimates of the number of species on Earth vary from 3 million to 100 million. And a more updated figure from an analysis of the IUCN's Red List of Threatened Species approximates that only 1.8 million species have been described from a total that ranges from 5 million to 30 million species. This implies that we have a lot of species that have barely been characterized and a lot more for us to discover. And at the heart of the importance of studying biodiversity is in the maintenance of ecosystem productivity. Basically, each species in an ecosystem has a niche, a role to play. Identifying the different species that we have helps us keep track of their existence and the roles they play, and perhaps also help in their conservation. Molecular biology has helped in the monitoring and conservation of biodiversity through the technology known as DNA barcoding. DNA barcoding involves the isolation and amplification of unique sequences present in the genome of different organisms. The unique sequences serve as barcodes that help us differentiate one organism from another, especially when there are no obvious physical characteristics that will allow us to do so. For example, scientists have described a new species of the Philippine false gecko, a genus found nowhere else on Earth. The new pseudo-gecko was described by comparing its physical features with existing literature and data, and then verified using DNA barcoding technology. But beyond DNA, biodiversity monitoring and species identification can also be done using proteins. And this is the case for the small-bodied hominid, called Homo luzonensis, that was approximated to have lived in the island of Luzon between 50,000 to 67,000 years ago. Difficulties were encountered when they tried to extract DNA from the bones and teeth that were recovered, and this is to be expected given the age of the specimen. But deep in the hominin's teeth, proteins lingered, and though they may be degraded, they are still identifiable. And when scientists analyzed them, the sequence of amino acids revealed the species of the organism. Molecular biology can also be useful in differentiating species for environmental management. For example, climate change and various anthropogenic factors have been triggering the population bloom of a strange starfish known as the crown of thorns. They are known corallivores that are considered the second major cause for coral loss, just behind coral bleaching. And in 2008, it was discovered that the crown of thorns starfish is actually made up of four morphologically similar species, as detected through DNA analysis. And the existence of multiple species could imply different modes of adaptation and therefore different modes of control. Meaning, if we are to prevent them from destroying our coral reefs, each species might require different types of management. The problem, though, is we can't yet differentiate the four species at a glance. And so our team at De La Salle University is hoping to change this by using DNA barcoding, image analysis, and environmental monitoring. My master's student Jovi Nicolas and our team seeks to identify the different species of crown of thorns present in the Philippines, and hopefully find morphological characters to differentiate one species from another using image analysis that is backed by DNA barcoding technology. You see, we've actually done something similar before, but more in the field of aquaculture. In the Philippines, we have 3 of the 4 known species of alimango, or mangrove crabs, under the genus Scylla. Out of the 3, only one species is preferred due to its faster growth rate and larger sizes upon maturity. This species is known as Scylla serrata. At juvenile stages, where the organisms are captured or traded for grow-out, species identification is difficult, if not previously impossible. But our team, as part of my dissertation a few years back, was able to solve this problem. I used DNA barcoding and image analysis to detect subtle differences in the carapace shape of juveniles to differentiate one species from another. This has since been developed into a free Android-based mobile application called Crabbifier that fishers and grow-out farmers can use. But how is Crabbifier connected to the environment and our climate change struggles? Well, prior to Crabbifier, fishers and grow-out farmers had no means to identify crablets to species. So, they would compensate by over-harvesting, which puts a strain in our ecosystems and also on the limited resources of our fishers. Crabbifier allows them to limit the number of samples they take from the wild, because they can verify their species, even for those that are traded. And they can also focus their resources on species that are more resilient to climate change. And that's the thing about climate change. It endangers existing systems for food production by the changing of the countless conditions that existing species thrive in. We are now racing against time to find resilient species and varieties, but molecular biology has also helped with this. Transcriptomics is the study of the transcriptome, which is the complete set of RNA produced by a cell or a tissue under specific environmental conditions. Through transcriptomics, we can study representative organisms of populations in different locations all over the world. The hypothesis is that different populations that have been exposed to different environmental conditions, such as high temperatures with low fluctuations or low temperatures with high fluctuations, would have developed different environmental adaptations. And we can verify this by exposing representative organisms to projected warming temperatures due to climate change. An analysis of the transcriptomes of a small number of organisms will allow us to figure out which populations can produce the right amounts of proteins, enzymes, and other molecules in order to survive and even thrive in different climate conditions. And once we found populations that show temperature resilience, we can also use molecular biology to predict their population structure. One aspect of population genetics helps us figure out the connectivity of different populations across geographic areas. In a nutshell, it tells us which populations are capable of exchanging alleles or genes because of interbreeding, migration, and other processes. In population genetics, we use DNA fragments and sequences to detect differences in what we call allele frequencies. And allele frequencies help us predict the patterns of reproduction and inheritance in the populations that we are studying. The more similar the allele frequencies are in two different populations, the higher the probability that they are connected. The more different, the higher the likelihood that these populations are isolated from each other. And this allows us to determine if stable populations can assist vulnerable ones, and also to help determine which vulnerable populations are interconnected, and thus adjust environmental management protocols accordingly. For example, our laboratory was able to determine that the mangrove crab species in the Philippines follow an east-west population genetic structure, as identified using microsatellites and mitochondrial DNA markers. What this structure suggests is that populations along the West Philippine Sea are interconnected, and so local government units must coordinate with each other in order to conserve and manage this organism. And the same is true for the populations facing the Pacific Ocean. And finally, we go to a very controversial molecular biology technique that can also help with our climate change battles. What I'm referring to is the process of genetic engineering. Through genetic engineering, we can alter the genetic programming of our crops to make them more suited to the changing environmental conditions. Genetic engineering, however, causes a lot of concern, some due to bad publicity, and others through concerns in control and destruction of biodiversity. Some GMOs available in the market are controlled by large corporations that have allowed them to monopolize certain markets. And since GMOs are better adapted to the environment, there are concerns that they will cause the extinction of naturally existing species due to the process of natural selection. But to clarify some of these concerns, let me first note that intentional genetic modification does not need to be done in the lab, and we have been doing it for thousands of years. And what I'm referring to is the process of selective breeding, the slow and tedious process of genetic recombination by mating two organisms that contain your preferred trait. Selective breeding has allowed the human race to flourish by allowing us to create new varieties of plants that grow bigger, grow faster, and contain more nutrients. Through selective breeding, for example, we have created larger and more nutritious corn. Selective breeding has brought about larger, tastier, and most importantly, non-toxic avocados. And without selective breeding, man's best friend would still be man's greatest predator. The process of selective breeding, however, tends to be slow and imprecise. When you do selective breeding, you have to wait for the natural fertility cycles of the organism in order to find out if your mating worked. Meaning, sometimes it will take months, years, or even decades to find out if your experiment was successful. And despite your time, your effort, and your investments, there's actually a huge probability that the desired trait will not be expressed in all the offspring, or even at all. And so, the precise and accurate process of genetic modification was invented. Genetic modification is rooted on how the DNA is the blueprint of life. Changing it can change an organism's physical characteristics. So, how does this work? Basically, small snippets of DNA are introduced into an organism to express a protein that will change its physical characteristics. We can create plants that are resistant to pests and viruses. We can even make it produce more of a healthy substance, such as a vitamin. And these traits are not artificial per se. They all come from nature, just from a different organism. And since the mechanism of understanding the DNA is more or less the same from a banana to a human, adding little snippets of DNA can help us change the characteristics of an organism. And so genetic engineering doesn't introduce chemicals or toxins to the organism. You just introduce an already existing piece of DNA that will be destroyed with heat or enzymes found in our saliva. And being a GMO is also not contagious. Touching them, being near them, or eating them won't transform you or any other plant or animal into a GMO. But in terms of reducing biodiversity, please consider that agriculture is already doing this by choosing only preferred varieties for growth. And so the reduction of biodiversity through agriculture will require a complete overhaul of our agricultural systems, not just the removal of GMOs as an option. And since most GMOs in the market are also designed to be incapable of breeding, then the issue of survival of the fittest won't be an issue at all. But given how patenting of GMOs is still under discussion, there are ways and means to make sure that only specific companies will have a monopoly on a specific GMO. And this is where the goal of helping the environment, creating more nutritious food, and producing enough so that world hunger is minimized can become an issue. We have to empower nonprofits and research laboratories to find the ways and means to make sure that our products reach the least, the last, and the lost, and so that it doesn't only benefit the few. In my experience, for example, we have used genetic engineering to create a variety of ABACA that is immune to the ABACA bunchy top virus. The ABACA bunchy top virus, or ABTV, is considered to be the most injurious of diseases in our local industry, which causes the weakening of the ABACA fibers and the stunting and eventual death of the plants. There are no available treatments for sick plants, and so we wanted to help by creating one that doesn't get sick in the first place. And this could potentially help our local farmers who have had to face problems such as the complete wipeout of more than 16,000 hectares of farmland over the 25,000 that is in existence in our country. However, the products of this research have remained mostly in the lab and testing fields due to lack of acceptance in our local communities. Farmers who dared to test the use of GMOs were scorned and vilified, to the point that some of them even suffered terrorism through the destruction of their crops. Right now, the scientific community is excited that golden rice will be planted in the Philippines again. This is a genetically modified rice that has vitamin A in it. But this has also triggered the anger and fear of a significant portion of the Filipino people. And at this point, we have to confront the fact that genetic engineering is a double-edged sword that can be used for good or for bad. The intentions of the users and creators will always come into question. And this has certainly become more of an issue now that the academe is being pressured to patent and to have return of investments. As uncomfortable as it is, the products of research, of science and technology will eventually have to earn in order for the systems that we have to be sustainable. And so we must look at genetic engineering as just one of the tools in our arsenal. It is not the be-all and end-all of any action towards climate change, but it is certainly one that has a lot of power. And that's it for my short discussion on select molecular biology techniques that can help in our battle against climate change. I hope I was able to give you a glimpse of what molecular biology can offer and how it can positively impact the world. If you have any questions, comments, or suggestions, I would be very happy to answer them. Thank you very much for this wonderful opportunity.